Hello, welcome you all to the Igniting Curiosity in Life Science. So today we'll move on to another very important the breakthrough or the concept in the biotechnology, that is especially what the genome editing, that is what the CRISPR Cas9, the concept. So this is Savita, Igniting Curiosity in Life Science. So without delay, let's dive into the subject. Okay, have you ever imagined editing genes like cutting and pasting text on your computer? Welcome to the world of CRISPR Cas9, the powerful tool that lets scientists rewrite the code of life itself. So, CRISPR stands for the clustered, regularly interspaced short palindromic repeats and CRISPR associated protein 9. In simple terms, it's a molecular tool that can cut, remove, or replace specific DNA sequences inside living cells. So, did you know that even bacteria have their own immune system? Yes, it's true. Bacteria also use a powerful defense called the CRISPR-Cas system, a fascinating form of adaptive immunity that helps them fight off viral invaders known as phages. Here is how it works, we'll discuss now. So, when a virus infects a bacterium, the bacterium captures tiny fragments of the viral DNA and stores them inside its own genome. These stored sequences act like molecular memories, a record of past infections. Then, if the same virus tries to attack again, the CRISPR system quickly recognizes the invader using those DNA memories. It sends out a special protein called Cas9, which cuts and destroys the viral DNA, stopping the infection in its tracks. So, yes, even the smallest organisms like bacteria on Earth have brilliant ways to survive. And even scientists have now turned this bacterial defense system into one of the very important, most powerful gene editing tools in the world, that is CRISPR Cas9. So, how actually it works? So, in step 1, what happens? Scientists create a guide RNA that matches the gene they want to target. In step 2, the Cas9 enzyme acts like scissors, cutting the DNA exactly at that particular spot. And in step 3, the cell naturally repairs this break. And during the repair, a new gene can be inserted or an old one is disabled. Okay, so why it is revolutionary? CRISPR is precise, fast, and inexpensive, transforming medicine, agriculture, and research. Okay, it's one of the biggest breakthroughs in modern biology. So, what are the main applications of this? So, CRISPR can fix the genetic diseases like sickle cell anemia, create pest resistant crops, and even help in cancer treatment. The possibilities are endless. Ethical concerns, but powerful tools come with the big questions, right? Editing human embryos, the risk of off-target effects, and designer babies. These are serious ethical concerns the world is still debating, right? So, this much about today's concept, a short introduction and a note on this. And let's discuss some more MCQs related to this the topic. So, first question, what does CRISPR stands for? It is stands for clustered regularly interspaced short palindromic repeats, right? And next question, what is the role of Cas9 in the CRISPR system? Guide RNA formation, DNA cutting enzyme, DNA repair enzyme, and transcription factor. Yes, that is the answer is DNA cutting enzyme. And third question, what is the function of the guide DNA? Sorry, guide RNA. That is even it is called as what the gRNA. Repairs DNA, acts as a template for new DNA, directs Cas9 to the target DNA, and synthesizes protein. The correct answer is directs Cas9 to the target DNA. And fourth question, CRISPR Cas9 was originally discovered in humans, bacteria, viruses and plants. The answer is bacteria. Which of the following is not an application of CRISPR Cas9? Correcting genetic disorders, producing pest resistant crops, protein synthesis, cancer research. So, the answer is protein synthesis. And the sixth question. The process by which a cell repairs its DNA after CRISPR cutting is called gene translation, DNA replication, DNA repair, RNA transcription. The correct answer is DNA repair. A possible risk associated with the CRISPR is off-target gene mutations, lack of specificity, gene silencing, all of the above. Yes, the correct answer is off-target gene mutations. And the eighth question, who received the Nobel Prize in Chemistry in 2020 for CRISPR-Cas9? Watson and Crick, Emmanuel Charpentier and Jennifer Doudna, Rosalind Franklin, James Doudna. Yes, the answer is Emmanuel Charpentier and Jennifer Doudna. And the ninth question, CRISPR technology can be used in agriculture to create genetically uniform crops, improve crop resistance to pests, produce chemical fertilizers, stop photosynthesis. Yes, the answer is improve crop resistance to pests. And the last question, what makes CRISPR more efficient than previous gene editing tools like Talens or Zedafens? So, it is less accurate, it requires a complex protein designer, it's simple and more cost effective, it cannot be used in humans. Is the answer is it's simple and more cost effective. Okay, so these are all about some MCQs regarding this topic. So that much about today's brief note on CRISPR Cas9. If you found this video helpful, please don't forget to like, share, comment and subscribe. This is Savita, igniting curiosity in life science. See you in the next video. Thank you.